Thank you very much. Hello, everybody. Before I can decipher this pretty long title, I would like to apply a couple concepts. Cancer is a highly adaptive disease. It avoids immune response very effectively with various mechanisms. One of these mechanisms is suppressing the immune system directly through so-called immune checkpoint molecules, which are located on the surface of immune cells. And the tumor is able to express ligands which directly bind these immune checkpoint molecules and therefore deactivate tumor killing activity. Like you can see in the image, that if this were an immune checkpoint molecule, the red dots bind it, which the red dots are the ligands, and this way there is going to be a signal be sent to the immune cell to deactivate its function to present the tumor to the killer cell, and therefore the whole immune system is being a lot less effective. So the LILRB receptor family is part of these immune checkpoint molecules, and the focal point of this presentation is LILRB4, which is has been under a lot of study lately. There's an antibody therapy which targets this molecule, and it's on a clinical trial, but it is for myeloid leukemia, so there's still a lot of study needed in other cancer types, for, in our example, colorectal cancer. So in our study plan, our main goal is to inhibit the function of LILRB4. And if we do that, then the tumor is not going to be impacting this signaling pathway, deactivate the immune cells which have this molecule. So to do this, we design in silico an antibody which can attach to the LILRB4 and not allow the light to reach you. And once we have this nanobody, we can test its ability in vitro studies. So why we want to design a nanobody is because it's a very small protein. And because it's very small, it can reach its target very effectively. And a nanobody is something we can design it to find the LILRB4 much more affinity than the ligand, thereby reaching the receptor first and not allowing the ligand to also attach to LILRB4. And this nanobody design pipeline is a protein design pipeline, which has, and protein design has benefited a lot from the creation of recent models. And in the phase one of this design process, we design a novel structure for the nanobody with a diffusion algorithm. And how this works is that we start from a template to the nanobody. Diffusion applies noise to it, and in a follow-up denoising step, we able to generate novel structures, which are not super different than the template, but still it results in unbiased novel structures. In a phase two, we add a sequence to the structure. We use a deep learning based tool, which has also been recently developed. And once we have the structure and a couple of novel structures, and for each structure we have a couple of sequences, we generate multiple tens of thousands of novel sequences, from which potentially are going to be good for us, but most of them are not going to be good. So we need a filtering step to filter out the non-working sequences. And we can do this very effectively at AlphaFold 3 which you might have heard about. It's also a very recent tool, and it's has a lot of advantages over AlphaFold 2 in relation of predicting folding in complexes. So here we have the receptor and we also have the nanobody, and AlphaFold 3 is able to predict folding of the nanobody in this context very well. So with AlphaFold 3, we can select the best working sequences that we designed. So the first conclusion here is that we generate a set of nanobodies which have the function to bind to LILRB4 and inhibit its function to suppress the immune system. So it's very great, but we have a second task in our project, and that is to identify which cells express LILRB4 in the colorectal cancer context. And this is a question because biology has a lot of variance. So gene expression in one context, one environment looks completely different than in other contexts. And the immune system and tumor interaction is especially complicated. And we need to validate in the colorectal cancer, where can we even find, on which cells we can find the LILRB4 molecule. So in the literature, we see that they have shown LILRB4 on the surface of monocytes, macrophages, and dendritic cells. But we need to look into this ourselves in the laboratory. However, that's this process, and we were thinking of doing a shortcut or side project of looking at this computationally from publicly available data, where we don't even have to generate the data ourselves, and we can quickly get some results in this question. And I used single cell sequencing data. It's great that this was introduced in the previous talk. It has a resolution which is great. That's exactly what we need for this task. And why we use RNA data. Ideally, we would use protein data, but that's very difficult to reach. It's not very available, and it's also hard to analyze. However, RNA data is broadly available. It's easy to use, easy to analyze, and we assume that the amount of protein in a cell is very highly correlated with the RNA amount of the same in the cell. And the data set that we used here is a pretty considering with 62 samples and 370,000 cells from colorectal tumors and normal tissues. So the hindrance that we ran into with this task was that even though we had information about all of us in this data set, transcriptomic information, but it's not trivial that what type of cells, what cell types each cell belongs to. That's not trivial to figure out, but we need a precise annotation to be able to complete our task. And therefore we used recently developed language model-based tool called Cell-to-Sentence, which has the novelty to it that it takes an input of sentence, which is basically the names of the most expressed genes in descending order from a single cell, and that you can see, for example, here, and without any numerical use from these gene names, it's gonna give us a label for our cell. And according to publication, it has a very good performance for this specific task. But this comes with a cost of it lost its natural language capabilities. It just gives us a label, and it's not going to argue why it chosen this label, and we cannot ask for further arguments from it. So we wanted to compare the performance of this tool with a language model, which we ask about this annotation. We can ask follow-up questions. So we devised a comparison scheme between these two models. We used the same prompt that you saw on the left slide to analyze a subset of 1,000 cells. Each LLM gives us a very different answer in terms of how it's formatted. DeepSeek usually gives us a larger sum, and cell-to-sentence gives us only the label. So with some language model, I compared these two answers for each individual cell and rated it between 1 and 5. And I summarized the scores into this diagram. And what we can see there is that in more than half of the cases, the two models gave us very similar answers. So they annotated the cells very, very similarly. Even DeepSeek, which didn't receive any specific training for this task. But we see that in quite a bit of the cases, 30 to 40%, we get some differences, like not sure how big, but definitely relevant differences. And because we know that DeepSeek has a good accuracy in this specific task, we assume that DeepSeek is less accurate in some cells. However, this comes with the trade-off that with Deep we can talk more about this question because it gives us arguments why it chose its cell type labeling. So it's definitely something that you have to consider upon using these tools. So once we seen that, we utilize cell-to-sentence to annotate all of our cells. And on the left side image, you see the two UMAP mapping of all of our data, transcriptomic data, where cell types which are similar cluster close together. And I highlighted the relevant cell types. And if we compare that to the middle image, we can already see that with the brighter colors, there is higher expression of LILRB4. And we see that mostly macrophages express this marker higher, and monocytes with purple also express it very broadly, which is consistent to the literature. That's kind of what we wanted to see. But once quantifying cell groups below the first two cell categories, we also see that plasma cells, dendritic cells also marker. And that's very interesting for us. We didn't expect that because the plasma cells come from a completely different lineage than the macrophages. So this has not been shown in the literature. It needs further validation, and it's an interesting result so far. So as to the second conclusion, when comparing language model performance on biological tasks is unstudied, but it's a very relevant question. And in our case here, we argue that there exists a trade-off between using a specialized and a general model. And I want to thank to the contributors, partners, and funding for this study, and of course, my colleagues at the Department of Biophysics and Radiation Biology at SMLS.
So is there any question for Péter?
Thank you for the talk. So you compared a specific model, this cell interpreting model with DeepSeek. What was did you have any specific guidance why you chose DeepSeek as the reference? This is the first question. The other is, have you considered fine-tuning a base model on your data, whether that might turn out better?
Yes, thank you. So DeepSeek was just used because of its good general performance, and it's a free model, open source model. Nothing really special choice there other than that.
Is there some public benchmark that was indicating that it is good or better than other models?
Basically, yes.
I see.
Exactly. And I haven't developed into training a model like that before, but it's definitely some direction that I would like to go. Also collecting more data and pre-training with that. So that would be definitely like one of the next steps.
Thanks.
Thank you.
Hey, thank you for the nice talk. I wanted to ask that you mentioned that you generated multiple possible nanobodies in silico, and then you validating them in the lab. I was wondering if you have considered evaluating the binding, like the free energy of these, or like, you know, using some computational methods to estimate these generated proteins.
So can you repeat maybe just the bulk of the question?
Yeah. So why evaluate these in the lab, and why not use some computational methods instead?
So this is just surface of the nanobody design pipeline in the in silico part, and we had more filtering steps, and we had a more iterative work to selecting the better binding ones. This is very much just the skeleton of it. We used multiple other tools than these. And in the lab, it's just going into the lab right now. So we selected 5 to 10, and they are going to the lab just as we speak. So that's basically where it's at.
Thank you.
Mm-hmm. 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 That's a great question. I know about the effect itself, but I'm not super sure. I can very much guess that it was considered in the design pipeline. So as I mentioned, that there have been multiple tools that a bit more iteratively down the line we checked for other effects. And I can't comment in more more detail. Thank you.